Hello, and welcome to a special ACR Convergence edition of ACR Journals on Air. I'm your host, Vicky Shamagam. Macrophage activation syndrome and autoinflammatory diseases drive fear into the hearts of all rheumatologists because they can be so abruptly devastating. Today, as part of our ACR Convergence 2024 series, we're joined by two incredible scientists working on systemic autoinflammatory diseases. Dr. Grant Schulert is an associate professor of pediatrics, a rheumatologist, and a researcher at Cincinnati Children's Hospital. He received his MD and PhD from the University of Iowa and completed his residency at Vanderbilt University Medical Center before moving to Cincinnati for fellowship and later joining the faculty. He specializes in treating autoinflammatory disorders, including systemic JIA and macrophage activation syndrome. And Dr. Arthur Kaser is a professor of gastroenterology at Cambridge University in England. His research has provided fundamental insights into mechanisms driving inflammatory bowel disease, and he's also studying T-cell priming and the role of dendritic cells in macrophage activation syndrome. Welcome to the show, Grant and Arthur.
Thank you very much. Thanks for having me.
Same here. Great to be here.
So you both are speaking together at Fueling the Flame, New Mechanisms in Autoinflammatory Diseases. And I should mention that the third presenter in the session, Dr. Jessica Hammerman, was unable to join us today, but will speak during the Convergence session. And her topic is toll-like receptors and macrophage activation syndrome. As we kind of frame this discussion today, Grant, I wanted to start with you. And I'd like to know if you could just give us a quick overview of refractory systemic JIA and kind of fill our listeners in on the framing for this session.
Yeah, thanks, Vicky. So the systemic form of juvenile idiopathic arthritis or systemic JIA, it used to be called Still's disease. And in fact, my adult rheumatology colleagues still call their form of this adult onset Still's disease. And there's actually a move to going back to calling this whole spectrum Still's disease so that we can have better nomenclature and have better conversations with our adult colleagues. Systemic JIA is one of the most distinctive forms of childhood arthritis because, as the name implies, along with joint problems and arthritis, they can have inflammation all throughout their body. And it's noted by these really prominent daily fevers, rashes, liver dysfunction, as well as risk for systemic complications, including lung disease and macrophage activation syndrome. And macrophage activation syndrome is sort of in this spectrum that we call cytokine storm syndromes, where diseases that are really driven by pathological and uncontrolled inflammation that's really blown past the brakes of normal inflammatory responses that try to fine-tune and regulate things in a controlled manner. And in MAS, you see really this overwhelming and catastrophic inflammation that's being uncontrolled by those normal regulatory systems. And so many children with systemic JIA do very well, particularly in our biologic era. However, anywhere from about a quarter to a third of them have a more complicated or refractory course, which means that they aren't having a sort of a complete response to the first-line biologic drugs that we use, or they're having significant complications like recurrent episodes of macrophage activation syndrome or lung disease. And, as I've talked about all of these different features that kids with systemic JIA can have, you can see the real clinical diversity we see in this disease, that some of them can really have mostly arthritis, some can have mostly systemic symptoms, they can have these various complications. It becomes a very clinically heterogeneous disease when we think about how to study it.
Tell us a little bit about your work studying the immune landscape of systemic JIA at the single cell level.
Yeah, so we're really interested in understanding what's happening in systemic JIA, particularly in these more complicated and refractory courses. What's different about the inflammatory responses in those children from those who have a more typical course of systemic JIA, which, as I noted, usually has a very nice response to biologics that block things like IL-1 and IL-6. Several years ago, we wanted to leverage the expertise of my colleagues here at Cincinnati Children's in doing single-cell biology to try to better understand what's happening rather than looking at sort of one particular cell population or sort of bulk studies in the blood, but really across that immune landscape. So single-cell RNA sequencing gives us the ability to from a patient to sequence and get transcriptional profiles from thousands of individual single cells simultaneously, and then understand what's happening in CD4 T cells, what's happening in CD8 T cells, what's happening in various flavors of B cells, what's happening in various flavors of monocytes, all simultaneously, and understand how each of those different immune populations is responding, and if they're responding in similar ways, if they're responding in different ways, how those are different from healthy control children, and how children with, as I said, more complicated courses are different from those with a more typical course.
Yeah, so it's a little bit like seeing the musical score for the whole symphony.
And giving you the ability to sort of tune out everything except that second violin and just being able to hear that. But then, of course, the challenge then is how do you integrate all of those different instruments and all of those different sounds at the same time to make sense of it and make it not just be a cacophony of sound.
Yeah, I guess that's what the amazing pediatric rheumatologist does as the conductor of the orchestra.
Some days, we hope so.
So I'm going to confess to our listeners that you sent me a little bit of a teaser about what you were going to be discussing during your session at Convergence. And you said, I'm going to quote, using UDON and SATAY-UDON to discover novel disease programs. So I did what any self-respecting scientist would do when confronted with a term I'd never heard of, which is I plugged it into Google. But the top hits I got back were all to do with noodles. Udon and SATAY-UDON are not, in fact, types of noodles or food, although I suspect the computational biologists were perhaps having some fun with the naming. What are they, and how are you using them to drive discovery for kids with autoinflammatory diseases?
Yeah, we were definitely hungry when we had that meeting. Actually, our team meetings, I think, were at 11 o'clock at that time. And so we were all beginning to think about lunch. And so we had a whole meeting to think about what we wanted to call these approaches. And they were all food-themed names. We considered tempura. My postdoc was really advocating for tempura, but udon made the most sense. UDON stands for Unsupervised Discovery of Novel Disease Programs. And so what we were interested in doing, and I should note that this was really a collaboration between a fantastic former postdoc in my lab, Emily Verweyen, who's back in Munster, a wonderful informatics PhD student who's getting ready to defend her PhD, Karabe Thakar. The two of them were working with us to try to understand how can we analyze the single cell data when all of these patients are so different, that some of them have new onset disease, some of them have established disease being treated by various medicines, some of them have this symptom or this complication or that complication. And particularly then when you overlay the thousands of single cells that we get. And so what UDON does is it takes the different cell populations that you find in single cell biology. So in this case, we found 27 different populations of cells in our patients and collapses all of those cells into sort of one group. And it's called a pseudo bulk. So it's like you did a bulk RNA sequencing experiment only on that one population. And so you get all of those 27 populations across all of our patients. And then what it looks to find is shared transcriptional programs or gene signatures across the different cell types and across the different patients. And without sort of any pre-existing hypothesis about whether this patient has active disease or inactive disease or this complication or that complication, and just sort of looks for patterns. And what it finds is these things called UDON clusters, which are groups of particular cell types in particular patients that have similar gene expression changes. To give one example, particularly in patients with macrophage activation syndrome or who had sort of early or what we call subclinical MAS, we found a very similar interferon-driven gene program across several different myeloid and dendritic cell populations, but only in those patients that had MAS or early features of MAS. And we found, I believe, 12 different programs across these different patients with these various complications. And some of them were things that we knew, like we knew that interferon was really critical for driving MAS. But some of them were things that we'd never really thought of. For example, we found a program that was present in platelets and platelet precursors that was really driven by some of these alarmin proteins, which are sort of innate amplifiers of immune responses, which we never really thought about platelets as drivers of the inflammatory response in this disease. But this approach sort of suggests that's a hypothesis that we should pursue. And so that's UDON. And then SATAY is a statistical test for association for UDON. So what it then does is takes all of the things we know about those patients. Are they boys or girls? Are they on IL-1, anti-IL-1 therapy? Or are they not on therapy? Do they have arthritis at the time of sampling or no arthritis? And then associates those various clinical features with those particular programs. And so it can say that, oh, okay, well, that interferon-driven program that we see in myeloid cells is in fact associated with these clinical features, these laboratory tests, to be able to sort of give some context to these programs that you can do. To show that we could do this in more than just systemic JIA, we then pulled a couple of publicly available data sets. There was a large lupus, pediatric and adult lupus single cell study that had been done by Virginia Pasquale and others several years ago. We pulled that, we ran it through UDON and UDON SATAY, and we found some of the same things that Virginia found. We found, again, this very strong interferon signature that we see in lupus, but we can actually, through a different mechanism than what they used, show that it's associated with markers of high lupus activity and in particular cell populations. Lastly, we took a really interesting data set which looked at the 1K1K, which was sequencing at least 1,000 cells from 1,000 healthy individuals. So it was almost 1 million cells from quote unquote healthy. I mean, who among us is really healthy, but
what is health?
Right, exactly. Who is normal, right? But 1,000, quote, normal people. And what it found was that, well, almost all of those cell populations clustered with sort of the normal healthy groups in our experiments. A few of them didn't. Something on the order of about 5% or so of people have immune populations that were clustering with some of the things that we see in inflammatory disorders. And unfortunately, there's no data on these people, but the question is, do these people have sort of early autoimmunity? We know from many studies that in people who develop autoimmunity, if you have the opportunity to look at their blood several years before they develop autoimmunity, you begin seeing these little flickers of autoimmunity. And so whether these are people who have sort of early inflammatory diseases. So sort of showing some of the power of being able to use tools like this for other clinically heterogeneous diseases like lupus or other rheumatic diseases that we're interested in.
That's really interesting. And I guess I have a few follow-up sort of pragmatic questions. Is this platform and code that you're running unique to your lab? Is it something that's widely available for other people to use?
Yeah. Great question. So it's on GitHub. It's easy to download and run as an R package. If you have any issues with that, shoot me an email. But it's available on GitHub and linked in our paper.
Great. That's really amazing, because I can imagine that many people will actually want to test this out. And I'm kind of interested to see where it goes next in terms of looking at other disease entities. Your work obviously is at the forefront of discovery in pediatric rheumatology. Can you give us a bit more detail about what you're learning about the transcriptional diversity of the normal density and low density granulocytes in systemic JIA?
Yeah. One of the other areas that we've become really interested in is looking at neutrophils in systemic JIA, because, again, this is a very sort of inflammatory and autoinflammatory driven disease where we think neutrophils play a really key role, but they're really understudied. And partially because they're kind of a pain to work with. Laboratory scientists will certainly know this. They don't last in the lab. You can't culture them. They don't like being frozen and thawed. For our UDON study, those were with frozen peripheral blood mononuclear cells, and so there weren't really any neutrophils in that. So another postdoc in my lab, Natsumi Inoue, has been trying to study neutrophils in patients with systemic JIA, both the normal density neutrophils as well as the low density granulocytes. And I'm hopefully going to be talking a little bit about some of this early data at ACR next month. But what we're finding, which is really interesting, is that low density granulocytes, it's sort of unknown how they're related to regular neutrophils in patients with inflammatory diseases. What we're finding is that they're sort of similar and different, right? That we see a lot of cells that, at least on a transcriptional level, look a lot like regular density neutrophils. However, we definitely see sort of an enrichment in sort of more immature cells, as well as cells that seem like they're more primed towards sort of netosis and other sort of pro-inflammatory behaviors. So we're really excited, and we've developed some kind of cool ways to study those on a transcriptional level that I'm excited to talk about, and I'm excited to see where Natsumi goes with it in the near future.
Oh, well, so you've teased us. So how are you using these approaches in new work going forward?
What we've been working with is a really cool system for capturing single cells sort of freshly and being able to preserve them for sequencing. And then Natsumi has sort of developed this approach where she can separate out the low density and normal density neutrophils from a patient sample and then sort of sequence them separately. And then we can compare directly from in a single patient, how are they the same and different, as well as then across patients through things like UDON, and associate how are the differences in these patients being able to be resolved in the neutrophil compartment as well.
Yeah, really cool. Arthur, I am going to turn to you now. You are going to be presenting on dendritic cells and particularly T cell priming in macrophage activation syndrome. Before we start on macrophage activation syndrome, I need a bit of background. You study predominantly inflammatory bowel disease. So how did you get from inflammatory bowel disease to macrophage activation syndrome? And then can you set the scene for us a little bit on why you think dendritic cells are important?
This is a good question. As you mentioned, I'm a gastroenterologist, a mucosal immunologist. And I need to step back a little bit and shine some light on a few decisions that we made 2011, 2012. This is the heyday of genome-wide association studies. We all of a sudden had these large maps of genomic coordinates that were linked to risk, with many loci actually are not being annotated functionally at all. And when you think about it, there are only two ways to generate mechanistic insight into human disease from human data where cause-effect relationships are really resolved. One is an intervention, meaning, for example, a clinical trial, this classically fulfilling Cox postulate. When you block TNF and have an effect, then you know that TNF is involved. And the only other route is germline genetics. Genes don't change. So we started in 2011 where we thought, okay, let's pick one particularly interesting gene amongst those that are linked to inflammatory bowel disease. As you say, this is my scientific as well as clinical background. And see if we, by digging deeply into one single gene, we can learn a new pathophysiology. And we picked one that didn't have a proper name at the time, was C13 or 31. And it intrigued us because a coding variant, so an isoleucine valine substitution, was linked to increased risk of Crohn's disease, so one of the inflammatory bowel diseases, as well as leprosy, chronic infection with Mycobacterium leprae. In addition to this, half of the protein had close orthologs in bacteria. And several of these bacterial enzymes, or proteins, I have to say, at the time it wasn't clear what it really was, had been crystallized and the structure solved in the early 2000s, and there was not really a single domain on these proteins that functionally annotated. We took a leap of faith, started with a blank sheet of paper to work on this, and whilst we were in the midst of it already, where there was a beautiful report showing that another ultra-rare variant, first discovered in offspring from consanguineous families, led to either a monogenic form of Crohn's disease or led to a monogenic form of systemic juvenile idiopathic arthritis.
Aha, the connection.
This is the connection, indeed. And by now, there are multiple of these ultra-rare variants described in the literature. And to my knowledge, this gene is still the single gene linked to monogenic forms of systemic juvenile idiopathic arthritis. So this is the link essentially that brought us here, as Grant has already alluded to. This risk of macrophage activation syndrome is an inherent feature of systemic juvenile idiopathic arthritis. And this, in the end, got us to dendritic cells as well as T cell biology. T cell expansion is a very critical part of the pathophysiology as it appears. What we did is we used gene editing to put these human risk variants, both those with Schult's disease as well as the common variant that is carried by about a quarter of the population, into the mouse genome. With the background of macrophage activation syndrome potentially being triggered by otherwise trivial viral infections, we infected these mutant mice with influenza, did observe that this protein operating in dendritic cells plays a very central role in the priming of the anti-influenza T cell response in the vigor that these T cells have into their role in tissue destruction.
Oh, that's fascinating. So you also found something interesting about purine metabolism. Tell us what role purine metabolism plays in macrophage activation syndrome.
Yeah, sure. So we had this orphan protein with no annotated function. And long story short, we discovered using completely unbiased approaches that this protein metabolizes purine nucleosides and nucleobases. So we had this protein, you need to imagine actually in this way, that was strongly linked to energy metabolism, inflammation. We had published this already several years back, but it was unclear how it could perform these various profound actions. So we devised a strategy to search for new catalytic activities of what we thought was most likely an enzyme. And in this unbiased approach, to our big surprise, the hits that came out, purine nucleosides, adenosine, guanosine, inosine, methylthioadenosine, and adenine. This was a very big surprise because central purine metabolism is universal. Adenosine are considered primordial metabolites from which life is thought to have emerged some 4 billion years ago from prebiotic chemistry. So here we are actually, our new technology that looked in an unbiased way for new catalytic activities, and we saw that this affects these purine nucleosides. And on top of that, 4 of the 5 catalytic activities that we discovered in this way had been thought to be the sole domain of so-called monofunctional enzymes, adenosine deaminase, purine nucleoside phosphorylase, methylthioadenosine phosphorylase, that are universal in any form of life. And then there was an additional one where adenosine and adenine was directly interconverted as something that was thought to be absent from higher forms of life, supposed to only happen in bacteria. What I'm going to present in the meeting is how we could then link these purine nucleoside metabolizing effects to dendritic cell biology and how this very mechanism controls at the most fundamental level the priming activity of dendritic cells. So what happens is that there is a purely biochemical mechanism that we uncovered that operates to control the priming activity via, on the one hand side, controlling cytoplasmic redox state, the NADH to NAD plus ratio, and at the other end, impacting on the release of inosine. And together, they set the vigor in which naive T cells, both CD4 as well as CD8 positive T cells, are being primed. To give you just an example how profound this is, if you happen to have the Crohn's disease associated variant, which we found to be a slightly less active variant compared to the fully active variant, and you are being exposed, or these mice actually carrying these human variants are being exposed to influenza virus, they develop 4 times more influenza-specific T cells, which secrete substantially more interferon gamma, so the key mediator of macrophage activation syndrome, much more cytotoxic, release more granzyme compared to those that carry the fully active variant. And as I mentioned, this is a purely biochemical mechanism. There is not a single transcript differentially expressed between those dendritic cells that have the fully active variant compared to the hypomorphic variant. And we have also put the human variant that is linked to monogenic Still's disease or systemic juvenile idiopathic arthritis into these mice. And their response, their T cell response that is elicited is even stronger actually than the hypomorphic ones, leading also then to increased tissue damage. So essentially there is a biochemical mechanism that we consider one of the most fundamental controllers of immune activation that operates in dendritic cells, and that seems to play a very profound role in triggering this overvigorous, if you wish, T cell response.
Yeah, that's really fascinating. And so it sounds like you might have found the magic light switch. I'm wondering, do you foresee this metabolic checkpoint potentially being a target for new therapies for this condition?
Yeah, sure. Great question. I think this is a very attractive target for therapeutic intervention. Not an easy one, but a very attractive one. There have around 20, 30 patients been described with loss of function variants in this gene. they don't really respond to any of the conventional anti-inflammatories. So it could be a very attractive orthogonal approach. We have a strong interest in developing therapies here. We've spun out a company to develop precisely that.
Yeah, well, I look forward to hearing more in the years to come. So normally in this section of the podcast, we'll ask a little bit about people's lives as scientists. But today I want to take things in a different direction and find out a bit about how you liked meeting. So we're going to do a rapid fire ACR convergence quiz. You are both very experienced at attending scientific meetings. And my first question is, and I'm going to pitch this to each of you in turn, what are you most looking forward to at ACR Convergence 2024? Grant, you go first.
ACR is one of the larger meetings I go to physically in both ways, and like its length as well as the number of people there. And so it can be a little overwhelming. And I'm a pediatric rheumatologist, and pediatric rheumatology is sort of like a little subgroup within the ACR. So the PEDS year in review is always kind of like a PEDS family reunion, where everybody gets there a few minutes early because there's lots of hugs and excitement from seeing people and former colleagues and trainees and mentors. Then we have a pediatric focused clinical and basic science year in review to talk about some of the papers that are particularly of interest to pediatric rheumatologists, as well as an opportunity to recognize our colleagues who've won ACR awards, as well as some awards sponsored by the American Academy of Pediatrics for fellows in training, as well as some of our senior colleagues. So PEDS year in review is always a highlight for me.
And Arthur, I don't know how often you come to the American College of Rheumatology meeting, but what are you looking forward to?
Well, indeed, I'm mostly in GI meetings as well as general immunology meetings. I occasionally go to rheumatology meetings. I look very much forward to that session in particular, but also essentially to meet many of my rheumatology friends there. And I think it will be a very stimulating meeting.
How do you both like to spend your time at national meetings? Are you spending time in the plenaries, pounding the poster hall? Are you more likely to be found chilling out with a cappuccino in the exhibit hall?
I'm a big fan of walking the poster hall. I think it's a great opportunity. The really exciting new research that's just bubbling up is not on the plenary stage, but in those poster halls. I have several trainees, clinical as well as basic science trainees in my group. I love walking the poster hall with them to introduce them to colleagues, people they've read their work and know them, but to be able to meet them in the flesh is always fun. So I always budget plenty of time to walk the poster hall.
And Arthur, how about you?
Yeah, same here, actually. I mean, these are the most beautiful interactions, and you stumble often across really exciting research. And speaking with people gives you a completely different perspective than reading about work and exploring essentially the boundaries of their observations. So I look really very much forward to, and less so for a cappuccino, but rather than a double espresso, actually.
Of course.
And more after the cappuccino, the evening beer after the last session is often the light at the end of the tunnel.
Happy hour is always a good thing. Part of attending these scientific meetings is meeting the people doing the work. Chance encounters are really part of the magic that happens at ACR Convergence. Who would each of you most like to run into at ACR Convergence 2024?
For me, actually, one of the people I'm most looking forward to meeting finally is actually our co-panelist, Jessica, who I've actually never met in person, despite knowing her work for many years and her being a mentor for a good friend and colleague of mine, Susan Canny, who's a pediatric rheumatologist in Seattle. I'm actually very excited to finally meet her in the flesh.
Arthur, how about you?
I hope to bump into Georg Schett again. There's a beautiful new therapies for SLE and many other rheumatological diseases that is expanding in the autoimmune space at the moment. And I think there is an interesting overlap in biology that we are looking at. Yeah, I look very much forward to that.
Brilliant. And aside from this talk that you're obviously all presenting at, if a mentor or mentee wants to have a chance encounter with either of you, where can they find you?
I'm usually on the floor for the poster sessions. I have trainees, I think, with them. And all of the poster days, I have trainees with posters. And I'm also, otherwise in the breaks, I'm often at the PEDS Community Lounge there. The last pre-COVID ACR, there was a famous donut wall at the PEDS community when they first started piloting these community areas. And we all hope that it makes a reappearance. So,
I remember it well, Grant. I remember it well.
It's taken on almost mythical proportions, I feel like.
And Arthur, how about you?
Well, find me in the poster hall, but perhaps the most effective way is to ping me an email to ak729@comeacuk.
Brilliant. I'm sure some people will be looking you up. It has been fun to hear all of your plans for Convergence. I am excited to hear where your science is going. And actually, as someone who is not a pediatric rheumatologist, I am now fascinated to attend your session. Thank you both.
Thank you.
Thank you so much, Vicky.
My thanks today also go to immunologists, computational biologists, and physician scientists who are unraveling the noodles to understand complex autoinflammatory conditions in both children and adults. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow @ACRoom on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcasts at acrjournalsonair.org or wherever you get your podcasts.